So for session 4, the topic is smart targets and multimodality of cancer therapy. So we have 2 talks before the lunch break, and the first talk is by Cecile Malardier Jugroot, and she's a professor of chemistry and chemical engineering and VP research at Royal Military College of Canada. And I have to say, I'm Canadian. I'm very surprised to hear that the Royal Military College of Canada is doing cancer research. And she's going to talk about the development of novel smart materials for stimuli responsive and active drug delivery in the treatment of breast and prostate cancer. So, Cecile, if you want to share your screen.
Thank you. So I will share my screen. And start the presentation. Are you able to see my screen? Okay.
Yeah, it works.
Okay, perfect. Perfect. So thank you, Jean-Yves. Thank you for this invitation to give this talk. And yes, we do all sorts of research actually at the Royal Military College of Canada. And today I will talk about one of the aspects of the research that we do in our group in the Department of Chemistry and Chemical Engineering. And we're looking at the development of smart materials. So we have 2 different avenues for our research. One is really on targeted drug delivery, and this will be the focus of this talk. And the second part is the development of new material for nanoreactors and environmentally friendly synthesis, for example. So we like to look at nature and try to be inspired also by the self-assembly and the confinement effect, so that you see in cellular environment, but we try to use that also for materials. And a link with some talks that we saw this morning is really the development of gold layers, for example, as a very active catalyst. So we've developed a layer that's 1 atom thick of gold, which is a few nanometers in diameter, around 50 nanometers. And we use that only on the material side. Maybe we should also use it on the drug delivery side as well, as we saw this morning. So I will talk about targeted drug delivery in my talk. And we learned also this morning that you have different ways of targeting cells. And the most common way has been with passive targeting, using the leaky vasculature of very fast growing cells. And this is what we see in this slide, where large particles, and when I talk about large particles, we're still in the nanomaterial, but they are able to actually leak through this leaky vasculature and being incorporated mostly in cancer cells because they are the closest. But we can also link the passive targeting with an active targeting strategy. And this active targeting can be, for example, using receptors that are overexpressed in cancer cell, and we can bring those carrier directly to the cancer cell that way. So this is an example with folic acid. Folic acid is, the receptor is overly expressed in cancer and different types of cancer. And this was a very interesting part for us because we saw that if we can target cancer cells but not be specific to a cancer, we might have also more impact later on. So we can add folic acid to the carrier that we develop to target cancer cells specifically. This also avoids side effects of the treatment, of course. And then there is also another way of targeting the cancer cells, and it's looking at the environment of the cancer cell. And we know that the pH around the cancer cells is usually slightly acidic. So if we can have a pH responsive material and carrier to bring the drug directly to the cancer cell, it would be extremely beneficial. So if we can combine those three targeting strategies together into one carrier, we could have a major effect. So this project was started with 2 students. So Summer, who did most of the work on the development of the drug delivery system, and Matt, who did specifically the simulation work on this system. And we developed a system where we have, so this is the nanomaterial that we use. It is a polymer that will self-assemble into a tubular structure. The interior is mainly hydrophobic. So what we want to do here is being able to carry hydrophobic drugs to the cells. And the exterior is mainly hydrophilic. The hydrophilic part can be modified, and so we can have a covalent bond here between, so here is the molecule that will target the cell, and this is folic acid. And we have a linker between the folic acid and this polymer. And the polymer is actually pH sensitive. So this polymer will self-assemble only at intermediate pH, so here around pH 7. As you go to lower pH, then it will disassemble. This polymer is also biocompatible. It is used in pulp and paper. And if you have kids, you know that they chew paper, and so you need to have biocompatible molecules on paper. So this polymer is polystyrene alt malic acid. It is soluble in water, and at pH 7, we will have a self-assembled structure. So just to summarize the approach that we chose this polymer, which is pH sensitive. We chose a linker, and I'm going to show you also how we selected the linker to go between the polymer and folate. When the pH is lowered, this polymer will lose its structure and deliver the drug directly. So the polymer, and I'll warn you, my training was in theoretical chemistry, so that was the first thing that we did. It was really looking at the polymer and looking at the effect of pH when the pH is lowered. How does that influence the structure of the polymer? And what we've seen is that this polymer, which has an angle between the first monomer and the second one here at pH 3, presented a completely linear structure at pH 7. How does that relate to the self-assembly? Well, when you are at pH 7 and you're looking at a complete polymer with different chirality along the chain, you realize that this polymer will always have a linear structure. So if you have a linear structure, it will be very easy to get a self-assembly because the entropic cost of having this self-assembly is very minimal. When you are at low pH, then you can have very different structure. And here I'm looking only at 8 monomer. Of course, we have a long polymer chain, so it's completely disordered. You will need to stretch the chain before the assembly, and the entropic cost would be too much, and so that's why we see this pH sensitivity. Another aspect of this polymer is that when we are at a low molecular weight, which means very short chain for the polymer, we will obtain a nanotube. And the interior is hydrophobic, the exterior is hydrophilic. When we go to a longer chain, we obtain shown here, we obtain a nanosheet, and the interior is also hydrophobic. The exterior is hydrophilic. So this was really important for us because we wanted to solubilize hydrophobic molecules in here. The gap in here is the same as the one that we saw in here. So then we chose a biodegradable linker. One of the linker that is mostly used for biological application is PEG, so the polymer that is here. We also wanted to look at DABA. As a linker between the polymer and the folic acid. And when we did our simulation, we observed that with PEG, we would lose the pH sensitivity of the carrier, so we chose instead DABA. After a few years of synthesis for summer, we were able to develop this carrier, which is so SMA, which is the polymer, DABA, which is a linker with folic acid. And we've been able to look at the pH sensitivity. So if you look at the hydrodynamic radius that we obtain with dynamic light scattering, you can see that this hydrodynamic radius will decrease as the pH decreases. So we were quite happy with that result. And you can see that this is with a high molecular weight polymer. With the low molecular weight polymer, we had the same trend. So after doing all this work and the synthesis, Summer came to me and said, what if this doesn't work with cells? And then we turned to Myron for a lot of help with this project. First, we were trying to see how it would interact with the cell. And we've used turmeric to be able to trace this polymer inside the cell. So we solubilize turmeric inside this polymer as a fluorescent tracer. So here is Myron, and Myron helped us with testing on PenC1 first, and also raw blue cells. So looking at the effect of this new polymer on cells that had overexpressed folic acid receptors. So when we looked both at the effect of this polymer with turmeric inside in raw blue cell and in PenC1, what we saw is that we see the aggregation of the polymer also close to the cells. And we saw that the cells are stained, and including the nucleus. So we were able to bring the turmeric also inside the nucleus. The cells were losing their morphology, and we saw some apoptosis in this process, and this was common for both cell lines. Then we looked at just the polymer without the turmeric inside, and we looked at the cell viability. When we saw the same thing with PenC cells and also with raw blue cells. And what we saw is that we don't see a lot of effect for this polymer on PenC cell line. And I'll ask you to keep that in mind because this was done so with a very large molecular weight, but on one cell line. And you'll see that this is actually cell line dependent later on in the talk. But this was the first test that we did, and it was really encouraging. Then we looked at chemotherapeutic agents, and we looked at different concentration of the polymer with putting the drug, which is paclitaxel, actually as an excess. So the entire polymer in the interior part of the polymer had excess paclitaxel, and the concentration was controlled by the concentration of the polymer. So you can see an increase in concentration, and also you can see the change in cell viability as time goes on, so 24 hours to 48 hours and 72 hours. And we compared that with a high concentration also of paclitaxel. And you can see that we have a very high efficiency when we bring paclitaxel within the polymer. Then we had even more help from Myron and his group. And I'd like to mention that this work was done, the beginning of the work by Ruby, then Bessie, and then Alexandria. So you'll see all of them in different publications that we had on this subject. So the first part was done on individual cells, and we saw that maybe looking at spheroids and how those carrier can penetrate the spheroids would be extremely important. And what you can see here, so this is untreated breast cancer cells, spheroids. This part is the breast cancer cells with SMA, but without the functionalization. So this is not the target that we wanted. So we don't have the active targeting with SMA here. And this one is a spheroid, but this time targeted. And you can see a very big change as soon as we introduce the targeting. And after 30 minutes of exposure, you can still already see a difference. When you go to 6 hours of exposure, you can see the penetration inside the spheroid. So this was a very nice result. And you can see here a schematic representation of the polymer actually entering the cell through the receptor-mediated endocytosis. So this was a really nice result, but at the same time, we realized that the shape was also important during that study. And what I mean by the shape is when you have a 20 kilodalton polymer, which means short chains, you will obtain a nanotube. When you look at the very large structure, you obtain nanosheets. So this change between nanosheets and nanotubes actually induced a change also in cell viability. And you remember that at the beginning, I said we didn't see any difference in PENC1. Well, in using breast cancer cells, we saw a difference. And we saw a difference also in other cell lines. And this is an example of the change in cell viability as a function of the concentration of the carrier, and only the carrier without drug. And so you see that it's quite stable up to a certain point in concentration with the 20 kilodalton. When we have the sheet conformation, you see a significant drop in cell viability. So we thought something is happening here, and we need to study it a little bit more. And so what we've done, we looked at the 20 kilodalton functionalized polymer in PENC1 and also in MDA MB231, so breast cancer cells. Here you see the results that we obtained for the small polymers, so the nanotube, and here you see the results that we obtained with the sheets. And this is a combination of different drugs. So here you can see the different drugs that we have. So drug alone, drug with the polymer, drug alone, again, drug with a polymer. This is a combination of two drugs and with a polymer. And so you see this decrease in cell viability as a function of the treatment. But what we've seen and what was really interesting is that when you go to the breast cancer cells, you see a very big difference, and especially here when you have the drug alone and the polymer. And when you compare this part when we have the small nanopolymer or the sheets, you see that the effect of just the polymer is seen also here and here. So the main difference was seen, of course, as I mentioned before, in the breast cancer cell compared to PNC. And then because we saw a big difference, especially for PNC, especially for breast cancer cells, we looked at different protein levels and growth protein levels, proliferation protein levels with the treatment of the polymer alone. And what we've seen is that we saw decrease in those protein level, proliferation level after the treatment, but only with the large polymer, which made us suspect that the polymer was actually interacting with this regulation of the proliferation protein directly in the cell, and specifically for cancer. And so I'm sorry, I realize that this slide is actually a little bit blurry, but what we've done is we looked at caspase and the evolution as a function of time when we had the polymer, so the polymer with the functionalization. And so you see the effect that it has on the viability of the cells. And we use also anti-folic acid and free folic acid in coordination with the polymer. And so you can see the effect of the polymer and the high molecular weight polymer. And we compared that with the polymer alone without the function. And then you can see that this effect on the proliferation protein of the polymer alone is not here. And so that means that this, the folic acid and the interaction of the folic acid with the folic acid receptor is crucial in the control of the proliferation proteins. So then we looked also at the mobility of those cancer cells when the polymer was present. And what we can see is that with this scratch test, so you scratch one part of the slab here with the cells, you see that if you don't have any treatment, the wound will actually close, and you can see so this mobility of the cells. Now, when you have the polymer that is treating the cell, then you see that this mobility is not here anymore. So you see that the treatment is actually affecting different aspects of the cancer cells. And we looked specifically at other proteins, and we looked at the effect of the empty polymer, so just the carrier, on S1 and Notch1. And what we saw is that with the treatment and with the polymer treatment, we saw a significant decrease in Notch1 and also in S1. So this is the way we see now the effect of this delivery system, and not just delivering the drug, but as well having an effect on the proliferation of the cancer cells. So we could be acting both on the proliferation of the cell as well as treating with the drug that is contained inside. So I'm just cognizant of the time, so I'll just skip the conclusion because I really want to thank the people who actually worked on this study and all the students and also Myron as a collaborator and funding agencies. So I would like to thank you for your attention, and if you have any questions.
Thank you, Cecile. I think I'll start with one question. So you beautifully shown that your FADB polymer can form nanotubes or nanosheets. But do we know whether once it enters the cells, whether it would have different conformation within the cell? Could this actually modulate the response of the different cell lines?
Yeah. So what we're suspecting, because we really see an effect, we're suspecting that we're keeping the structure just because of the interaction with the cell when we're looking at the size of the cell and also of the receptors. And if you have a flat structure, it will be easier to actually have a contact with more folic acid receptors. It's a very complex question to be able to actually see it because of the scale, and also because when it interacts with the cell, then it will go under endocytosis. It will lose its shape, and so this is very difficult to have just a definite answer on that point specifically. Indirectly, because we see an effect. This effect, I think, is coming from the shape directly.
Is it possible to design such drugs so you have a particular static shape and then and see the effect on the cellular response?
Oh, can you reiterate? I'm not sure that I heard.
I'm just wondering if you could form, a particular drug that would form a static nanosheet. So you know that it's always a nanosheet and it's not a nanotube, but there's no dynamic between different shapes.
Oh, so between the different shapes, we know that there is no dynamics because we've been able to isolate them. Yes.
Okay. Thank you.